that this presentation, this whole workshop will be helpful for foundations that are starting out and really have to make a lot of decisions on which paths to take, and also foundations that are already underway and have some idea, but maybe we will be opening other horizons for them or enabling some decision making as well. I won't spend too much time on the business of science. This is what I do on a daily basis. I'm part of the business development team at Ultragenyx, which is a company focused on rare genetic diseases. I just wanted to focus on two topics that come up a lot when we speak with patients and foundations. And the first, which is actually really important, is how should you be providing funding to an academic? Should it be a grant or a sponsored research agreement? And did you even know that there's a difference between the two of them? The grant mechanism is usually very simple. It takes less time, which is always more attractive. It's usually cheaper because you don't have to pay for a lot of the overhead costs that universities usually charge when we put in place a sponsored research agreement. However, it comes with less strings attached, which is sometimes advantageous, but many times not, because you don't have any rights to anything that's generated out of the research that was funded. And in some cases, you may not want it or not need it, but you need to be aware of the fact that if any inventions come out of the work that you funded or any important information is generated, you don't necessarily have rights to it and you can't really do much with it. So in some cases, it's not an issue at all. You're just happy to get someone working on your disease and understanding the biology better and maybe publish so that it's available to others. But in cases where there is a therapeutic option and you want to control the fate of this therapeutic, you may want to look at the sponsored research agreement route. It's more expensive, as I said. You're paying for more infrastructure in the university, but you can get rights to any patent or intellectual property that was derived from the research and access to the data. So it takes longer, it's more expensive, but you get a lot more in return. So definitely when you are starting discussions with an academic, make sure that you understand what the end result of this research would be and what you would like to do with it when it ends. So that's just something that we've heard over and over from foundations that provided grants and then did not have any rights to the data, and the physicians either license the IP to somebody, or the academics license the IP to someone who didn't advance the science, and just lost the rights to the foundations lost the rights to it. And then 2 words on whether patents are important. So if you want to work with a company, yes, a patent is important and highly recommended that the academic or the foundation, if the invention comes from the foundation, that you file a patent. Costs money, I know, costs money to maintain it, but highly worthwhile because it gives you a strong position when you're talking to pharma, biotech, or other partners that might want to continue development of your inventions and take them into the clinic. There is a regulatory pathway called orphan exclusivity that sometimes can bypass the patent because the regulators will give you this exclusivity in the marketplace based on the fact that you were there first with an invention that no one came up with before. But the combination of having a patent and orphan exclusivity is usually the strongest for any company that wants to market a drug for a specific indication. So just the highlights, and obviously the discussions are much longer. I actually really like the definition of building a toolbox as a foundation when you're starting out. We see a lot of foundations that have that come to us and they have the toolbox on the left-hand side. They collected things from all over the place, not in an orderly manner, and don't really know what's in their toolbox. On the other hand, the right-hand side is a very nice and orderly toolbox. I don't have that one personally, but I'm sure people do. And it allows you to build your toolbox more systematically. If we can go to the next slide, Rodney. So what is a systematic approach to creating this toolbox or inventory list of what's inside your toolbox? So when you're starting out as a foundation, first of all, you need to understand what's already available. I mean, are there cells? Are there mice? Are there organoids? What has already been done, and where has it been done? And do you have access to it? Now, once you have that list, you start identifying the gaps in it. So if there are cells, but there isn't an animal model, then this is something you probably would like to focus on. If there is an animal model, but it doesn't necessarily, it's not the best animal model for a gene therapy, then maybe you want to consult with Ralph or somebody who's a gene therapy expert on maybe you need a different mouse. Who will make it? Will it be an academic lab? Advantages, disadvantages of working with academia versus a company, price, timeline, publications, etc. What would be the price? How long will it take? How do you, and as I alluded to previously, how much control do you have over the process, oversight, milestones, payments, etc., and who owns the results at the end? So I don't know that we need to go through all the lists here and every point. For each tool that my colleagues and my fellow panelists presented previously, there's obviously a decision to be made, whether it's needed, whether it's relevant, and how much to invest in it. Obviously, if you're just starting out and you're not a very wealthy foundation, you need to focus your efforts and decide what the most important things are that you need to start your journey and get research going and get people interested. If you have abundance of resources, you will probably try to do more. But there are things that take longer. Mice take longer than cells. A natural history study takes longer than a registry. So you also need to put things on a timeline and understand where you are now and where you want to be in a year or 2 or 3, and then start things that take longer, right, when you're kicking off things versus waiting, and then it will take a lot longer to get the information of the day that you need. We always recommend to make cells. We always recommend to have an animal model, but again, it's a matter of resources and timing. You go to Rodney and you ask him, which animal model should I make? Or what's the best animal model for my disease, my gene? Do I need more than one model? Do I need a knockout or an inducible model? And you know, Rodney will explain to you exactly what each is. Does the model have to be 100% identical to what the clinical presentation is? Is it even possible? And what happens if it's not possible? And then biomarkers are important, especially preparing your biomarkers towards clinical development. In diseases that the endpoints take a long time to follow, a neurodegenerative disease, for example, or a fibrotic liver disease, you want to see if the drug is working sooner better than later. You can't wait years and years to see a difference. So you want to be able to follow specific biomarkers, whether they're blood biomarkers or an imaging biomarker or an EEG biomarker, depending on your disease. You want to be able to establish this as a good biomarker for your disease so that it could be added into the clinical trials. And this will be done way before you go into the clinic. And another thing that we recommend, and I think this is maybe the strength of the foundations and the communities, is really know your community and know what a company like Ultragenyx or any other company needs to know about your community in order to help you develop a therapy. How many patients have been identified? what are the sexes, the age distribution at diagnosis, how many different mutations are represented. Is it, as Ralph said, if it's only one very prevalent mutation in most of the community, then there might be a gene editing approach. But if each patient has their own unique mutation that all leads to the same clinical presentation, then the therapeutic approach will be different. What are the most common clinical symptoms? And as a company, at least at Ultragenyx, when we start thinking about running a clinical trial, the first thing that we will do is survey the community and try to understand what each family and each patient actually feel are the most important things that they would like to see corrected or changed in their disease, so that there is a correlation between what we think is measurable to what you think is significant. Develop a registry if possible, and this is usually easier, and there are many companies these days that can help you develop registries. And more complicated and more time consuming and more resource consuming is the natural history study, and the difference between them is the registry usually captures only single time points, while a natural history study looks at a disease over time. And the reason that we as a company, and other companies as well, prefer a natural history study is that in many cases we can actually use the natural history study as a comparator group in the clinical study, and this saves time, saves resources, and also allows to run a smaller trial with less patients, which will be faster.